Hello guys, welcome back to my channel. Today I will explain about first let me say I will make some playlists in this channel. One of those playlists will be histology and cytology. Histology, it is the normal appearance. You will study the normal tissue of bodies, the normal appearance and structure, not the pathogenic. At your third year of vet college, as I studied in my country, is pathology, that you will study the abnormal. For example, cancer, inflammation of tissue, necrosis, and more. We have more and more. Okay, failure. You'll study those. Okay, we have four types of tissue: epithelial, connective, muscular, and nervous tissue. Okay. Basic histological techniques we have for tissue preparation. We should prepare the tissue before examining, of course, under microscope. Tissue, we have two types of microscope: electron and light microscope. Light is mostly used in histology labs. Okay. Steps required: first, fixation. When the tissue is received, okay, it's put in fixative materials. After putting in a cassette in fixative materials such as formaldehyde. Okay, dehydration will be done by alcohol in different concentrations. Okay, clearing will be done by xylene. Embedding the tissues will be put in paraffin and put in molds, or okay, or blocks, we can say. And embedding, as I said, mounting and cutting. Okay, mounting and cutting will be done by a microtome cutting. Okay, small sections will be put in water bath. After that, simply that I will make another video about staining. Okay, it will be more complicated. This is simply staining, just simply, basically. And after staining, we have the most common type. We have a lot of types of staining. We have trichrome stain, acid shift period stain. We have H&D. So H&D is the most common used type of stain. We have silver stain. Okay, mounting and cutting. After that, staining the section. Staining is, as I said, we have a lot of types. Here we have a simple figure. We will take a biopsy, and we have a lot of types of biopsies, guys. Be careful. We have FNA, or fine needle aspiration. It's just done by using a needle. We have surgical process. We will remove an organ, piece of organ, or an organ fully is taken to the lab. Okay, and it's cut into small pieces, and it's fixed by using formaldehyde. After that, it's dehydrated by alcohol, and it's cleared by xylene, and it's embedded. After that, it's put in paraffin block. Paraffin is added, of course. After those steps, sectioning with microtome is done. This wheel, as you see, you will mount it. You will put it here. You will put the cassette here. You can say the block, sorry, the mold. I mean, after that, you will just there's a wheel structure like at the right. You will turn that wheel, and you will cut small sections that the thickness is about 4 to 10 microns. Straightening, water bathing the tissues. Water bath is done. After water bath, this water bath is used to remove the paraffin wax, of course. Drying is done on a warmer. After that, staining is done. After that, yeah, as I said, staining is done. After staining, it's a cover slip is put it on a slide by using a DPX, and a cover slip is put it on the slide, and you will observe under the microscope in different lenses. Okay, by using different lenses. Cytology simply is the study of cell. Cell is the basic unit and structure of function of a living thing. Okay, we need cell. Okay, cell is the first step to form tissue. For example, what I mean is, a group of cells form a tissue, and a group of tissue form organs such as stomach. A group of organs form an organ system such as digestive system. Okay? Cell, a mass of protoplasm surrounded by a membrane containing a nucleus. We have a nucleus in every cell, and we know that cells are different in sizes. I will make this big paragraph simply. I will try to explain. I will try my best to explain it simply. Cells are different in size, such as have some cells are very small, such as the neuron cells are 4 microns only. But some cells are large, such as the ovum that's released from the ovary. It's about 120 microns, and we know that 1 millimeter it's 1000 micrometer. Okay, and there's an important note to know. There's no correlation between the size of animal and the size of its cells. For example, the cells of a mouse and the cells of an elephant are the same size. You know, they are the same size, but they are not the same amount. The amount is different. Okay, the cell are the basic functional, as I said, and they have different sizes and shapes. It may be oval, it may be cuboidal, it may be squamous or star shape. Okay, containing cytoplasmic process. Every cell contains cytoplasm. Every cell. Protoplasm is a living substance. Includes the cell components and the cell. And we have cytoplasm. The intracellular matrix extends the cell membrane to nucleus and contains the organelle and inclusion. Okay, the organelles are living. We have organelles are living and they do the metabolic activity such as cell membrane, rough endoplasmic reticulum, smooth endoplasmic reticulum, or RER and SER, Golgi apparatus or Golgi complex, mitochondria. Okay, mitochondria is one of the most important organelles, lysosome and centrioles. Inclusions are non-living, such as glycogen, lipid droplets, pigments, and microfilaments, microtubules. Okay, the nucleus is main fundamental. We know the genetic material is in the nucleus. DNA is in the nucleus, or deoxyribonucleic acid is in the nucleus, and direct synthesis of proteins and polypeptides known as transcription. This process is called transcription. Okay, the nucleus is usually spherical and it's centrally located in the cell. Most of the cells, the nucleus is located in the center, but you may get a pap smear or a vaginal smear. You may not find the nucleus of the epithelial directly in the center. Okay, that's an important note. It's not always in the center. No, that's not a deal. That it should not be always in the center. But most of the cells are the nucleus is located in the center. Okay. In some cells we have single nucleus, and in cells such as osteoclasts we have more than one nuclei. Okay, we have some cells nucleus may be elongated or different in shape, such as kidney. It may have a kidney-like shape or lobulated. Okay, the nucleus contains chromatin. Okay, the chromatin is in the nucleoplasm. Okay, we know that the nucleus contains a nuclear envelope by two nuclear membranes. The outer, it's continuous with the rough endoplasmic reticulum. So the outermost membrane of the nucleus, we have two membranes in the nucleus. The outer membrane, or I mean the first membrane, is attached to the endoplasmic reticulum. Okay, to the rough endoplasmic reticulum. I will tell you why it's called rough ER and small ER here, or smooth ER here. Where were we? We were here. There are nuclear pores that cause exchange between the nucleus and cytoplasm. Nuclear pores are just like holes. We can call them just hole-like structure that cause exchange of substances between the nucleus and mostly protein between the nucleus and cytoplasm. The nucleus contains chromatin, as I said, in the nucleoplasm. The chromatin is complex of DNA and protein. This chromatin is composed of the complex of DNA and protein. According to the degrees of chromosomes and condensation, they are divided into two types: heterochromatin and euchromatin. Heterochromatin is an active form, is inactive form of chromatin, dense aggregate located in the periphery of the nucleus, being relatively coiled, appears dark. The heterochromatin appears dark, but the euchromatin is active, and the DNA and the genetic material is in it, and transcription occurs in the euchromatin, and it appears mostly, it's just like translucent, and it is not dark when you see it under an electron microscope. Nucleolus is a spherical, deeply stained, non-membranous or non-membrane bounded structure composed mostly of the protein, RNA, and small amount of DNA. In light microscopy, you can see nuclei. Nuclei appears more basophilic. So what's basophilic and acidophilic? In H&E stain, we have hematoxylin and eosin. The eosin that takes nearly a red stain or a light stain, that's called any part of the cell that takes the light color of stain or red color, that's called it takes the eosin, or it absorbs the eosin, that's called the acidophilic. But basophilic appears dark under light microscope. So basophilic, this called morphology of the cell, basophilic, such as the nucleus, appears dark purple. Okay, if you see a dark purple, that means, such as the nucleus is basophilic, and the nuclei is basophilic too. As you see here, as I say, there are some holes called what? Nuclear pores. Okay, it's written here, nuclear pore. Here is the nucleoli. Okay, the nucleoli. This round big is the nucleus, and this is, you have two membranes and pores. Euchromatin is translucent, as I say, and the genetic material and transcription occurs here in the euchromatin, as you see here. But the dark black one is heterochromatin, okay, and it's inactive. Cytoplasm, simply, it's a jelly-like substance in the cell, which most of the chemical and metabolic activity occurs. Chemical reaction and metabolic activity occurs. And you have three regions or zones, we can say, are cytocentrum, endoplasm, and ectoplasm. Cytocentrum is next to the nucleus, okay? Next to the nucleus consists of narrow gelatinous region of organelle, except for endoplasmic reticulum attached to the nucleus envelope or a pair of centrioles. Okay, endoplasm adjacent to the cytocentrum, it means it comes after that. Okay, it's just like a small circle and bigger circle of cytoplasm. Okay, imagine it like that: small, bigger, and the biggest. Okay, draws three circles, in the middle of them the smallest, and after that a little bit bigger, and after that the biggest. Okay, the biggest one is the cytocentrum. Sorry, it's the ectoplasm. I mean, the endoplasm is, as I explained now, ectoplasm external to the endoplasm, and it's gel-like, and there are few organelles there. Okay, endoplasmic reticulum. It's a cytoplasmic structure, anastomosing network and sacs formed by continuous membrane and encloses a space called cisterna. Okay, there are two types of membranous system: rough and smooth. So rough ER, we have smooth endoplasmic reticulum and rough endoplasmic reticulum, RER and SER. Okay, smooth, it's called smooth due to the lack of ribosome. It's called the rough ER. It's called rough due to the presence of ribosome. So one of the most important difference is rough ER has ribosome in the outer membrane, okay, attached on it. We have free ribosomes and attached ribosomes in the cell. Smooth endoplasmic reticulum does not have any ribosome on it. It's completely smooth. Okay, the function of smooth ER: detoxification and producing steroid hormones. Okay, For example, in sperm cells, we have a lot of amount of smooth endoplasmic reticulum. Okay, because okay, the smooth endoplasmic reticulum is responsible in producing steroid hormones, phospholipids, carbohydrates, synthesis, detoxification, and storage of high level of calcium. Okay, rough ER is synthesis of proteins. Okay, lysozymes, a type of protein. Okay, mostly synthesis of protein. That's simply Golgi complex or Golgi apparatus. It's a tubular network that has two faces. This is an extra information: cis and trans face. Okay, and flattened cistern. Okay, found near the rough endoplasmic reticulum. It's responsible for generation vesicle that will transport nutrients. Cytochemical methods shown the Golgi apparatus present different enzymes. One of them is vitamin C synthesis occurs mostly in the Golgi apparatus. Lysosomes are membrane limited, so we mean that the first point, lysosomes are structures that don't have membrane. They contain enzymes and they are lysosome. Most of the scientists say, and it's already like that. Golgi, it's separated from the Golgi apparatus, okay? Lysosome, and it causes intracellular digestion. For example, any part of the cell dies, the lysosome will digest it. Simply, this is one of the example. Okay, mitochondria, it is considered a source of energy, or we can call the mitochondria a powerhouse. Power is produced in the mitochondria. Okay, the mitochondria possesses a smooth outer layer. We have two membranes. So mitochondria is a membranous organelle. We have two membranes in it, in the mitochondria. Okay, this is important to know. But I will explain the structure. Mitochondria is very important. Respiration occurs there by using glucose. And okay, the respiration occurs in the mitochondria, and I will explain it now simply. Most of the toxins affect the mitochondria, the cell. For example, cyanide. Okay, we will come to some important notes uh, sorry guys, to look at is which, sorry, just wait. Welcome back. So here, rough endoplasmic reticulum and smooth endoplasmic reticulum under electron microscope. You see those dot-like structure. Those are called the ribosomes. So it's called rough ER due to the presence of ribosomes. You will get those slides in the exam. Okay. Endoplasmic reticulum, smooth. You see, you can't see any dots in the membrane, in the outer part. Okay. Let's go to the Golgi apparatus. As you see, Golgi apparatus, the first face that I will magnify on it now is called cis face. Okay, this face, this first part, face. Okay, this face is called cis, and this is called trans face. Okay, the Golgi apparatus. This is the shape of Golgi apparatus, and it's easily, as you know, seen. So this is called the Golgi vesicle. This small thing, okay, small round-shaped things. Those are called the Golgi vesicles, or you can call them lysosomes. Here is a simple H&D stained cell. This is the cell body. This is then what's acidophilic and basophilic. The nucleus is, stains darker than the cytoplasms, you see? So that's called acidophilic. The nuclei or nucleus is is basophilic. So the nucleus, the nucleus is basophilic, so that's why it stains darker. The cytoplasm is eosinophilic, as you see here. As I said, the nucleus may not be directly in the center, as you see in this slide, in this tissue. okay? And this is the plasma membrane, round shaped, okay? Here I showed you a different selective stain called a silver stain. In silver stain, you see this white substance, this white round substance, this is the nucleus, okay? This stain is used to see Golgi apparatus. So as you see, those black hair-like structures are the Golgi apparatus. You can see the Golgi apparatus in light microscope only by using this stain. So this is called the silver stain. This is the structure of mitochondria, okay? We have the inner membrane and outer membrane. We have a special DNA in the cytoplasm, okay? Those dots, dot-like rounded structures, small ones, are called the ribosomes, okay, present in the mitochondria. These are the cristae, or okay, as you see those folds. So the last thing to know, you can have a rest between those explanations that I will do. Have a rest, drink a water, and come back, okay? Most of the adenosine triphosphate, or ATP, is synthesized during glucose metabolism. That's my cat. ATP synthesis during glucose metabolism is produced in the mitochondria through oxidative phosphorylation. So respiration, this is respiration, we can say. ATP synthesized during glucose metabolism. So, okay, that's what's called uh, simple respiration and oxidative phosphorylation. So thank you. Hope you get benefit. Any questions? I can answer it in the comments.